may know that he's been trying to travel back to Paris, where his family has remained over the times that he's been here for many months due to COVID-19. And he finally got, well, he told me that, and some of us who talk to him every week, that it's not a problem getting there. The problem is getting back to the U.S.
Yeah.
And so he said he's pretty sure he can get back. He's taking a chance, but at this point he really needed to see his family. So he's actually flying today. That's the flight that he could get out. So he asked me, and for those who don't know me, Wayne Guida, to introduce Dr. Baker and Nicole. And we do this pretty informally. So I'll start with Dr. Baker. Bill Baker is a good friend and a colleague. He's been at USF since 2001. And I've been there since 2007, Bill. Can you believe it?
Yeah.
It's been that long. But before that, he was at FIT, Florida Institute of Technology. He's a chemist, a natural products chemist, focusing on marine natural products. His lab is probably most famous for Palmerolide and now Darwinolide. Will, I think, rise to the same level. They go down to Antarctica. I guess you've gone almost every year, right, Bill, since you've...
It's actually works out to about every other year.
Every other year,
on
yeah.
average, yeah.
Yeah, and he usually takes at least one student with him. So that's a big attraction for students who have joined his lab. Nicole has been with Bill since 2000 and I don't have it in my notes, Nicole, 2016. Okay, all right, okay. Before that, she was a physician's assistant. She completed a BS degree at USF in 2006 in biomedical sciences. That's also part of our family because biomedical sciences, most of you know, but maybe some of you don't know, is housed within the Department of Chemistry. She then received a master's degree in as a physician's assistant. By the way, she had a minor in public health as well for her BS degree. And so she's currently working on her PhD in Dr. Baker's lab, expected to finish in December. She's already defended her dissertation. It was fantastic. She's, I don't hesitate to proudly say that she's one of our best graduate students. I've served on her committee. And so without further delay, I think Dr. Baker's going first and then Nicole.
Indeed,
so, yeah.
Indeed, right. And I'm just going to start sharing my screen. My purpose today is really to introduce what we do in my lab. This is a microbiome club, and we are chemists. And so I want to introduce microbiome club members to chemistry and the chemistry of natural products. If I can get my screen to work here.
It's up on my screen though.
The PowerPoint's not running though, right?
It was, well, yeah, it is.
There we go. That's what I wanted to see.
Ah, okay.
Okay, so, right. By way of introduction, how organic chemists might have an interest in microbiomes and the whole world of microbiology. I'm going to show you a few slides here just to orient the non-experts into the nature, if you will, of natural products chemistry, and as I say, how the microbiome plays a role in that. The project I'm going to talk about is highlighted here on this first slide. The title is a mouthful. I'll let Nicole tell you more about the details of that, but I want to emphasize that this is a team effort, and it's quite a huge team, as you can see just by all the names. But it's not just names that make it huge, it's disciplines. And so my own group with Nicole and two colleagues you see here, we're calling out, Sophia and Ryan, contributed.
So Bill, I see like a green bar that below the slide, and all I see is the title.
Really?
Yes.
Let me try again.
If you have to do it without going to the slide view, or just the regular view. Yeah, see, I can see that now. As soon as you went, yeah, I can see everything now. So if you need to just stay in the editing view or whatever it's called.
Yeah. So let me do that. Let me just stay there. See if I can shrink that down a little bit. Yeah, so we'll just work from here. Right, so not only is it a large team people-wise, it's a large team expertise-wise. So my own lab, as Wayne pointed out, we focus on natural products chemistry and drug discovery. Allison Murray at the Desert Research Institute and her team are microbial ecologists, molecular microbiologists. Allison and I met many years ago in Antarctica, and that sort of spawned this collaboration that we're working on, that we're going to tell you about today. Allison brought in Patrick Chain, eminent scientist at Los Alamos National Lab. Patrick himself is a microbial ecologist. His expertise that he brings to this project is in genomics and bioinformatics, and he has a wonderful team of collaborators there at Los Alamos, some of whom we've called out on this slide. But this is the team that's contributed to this. It's by no means just Nicole and I working on this. And in fact, the microbiome part that perhaps some of you are going to be most interested in here is really the work of Allison and Patrick. Allison is on the line here today, so if you all have questions that we can't answer, I am happy to say that Allison is in the wings, waiting to sort of talk you through some of what we've contributed here. So, right, my purpose is to introduce natural products to the microbiome community. So let me just start here. As Wayne said, this is a project we've been doing in Antarctica since the 1990s. It's primarily focused on chemical ecology. And there's my pointer. Chemical ecology is sort of what drives the program in Antarctica. And this is an interaction with the group at University of Alabama at Birmingham, where again, my group is interested in the chemistry, and the Birmingham group brings in the chemical ecology, the bioactivity parts of this work. This is just sort of a schematic of the workflow. We spend a lot of time in the field, making observations, scuba diving, collecting samples, doing biological screening in the field. The point is to discover new and known metabolites in marine invertebrates and try to understand their chemical ecology. Sort of offline, once we get back to the University of South Florida, we're also interested in applications of those metabolites, and I'll tell you a little bit more about that in a couple of slides further along here. So let me just say again for the uninitiated why chemists should be interested in microbiomes. So Nicole in her title has called out Palmerolide here. This is a natural product. I show the chemical structure of Palmerolide here. It was isolated from this Antarctic Ascidian. This is a marine invertebrate that, as you can see, is attached to the seafloor. And so the concept with chemical ecology is that these invertebrates can't flee from predators and whatnot. And so a lot of times they make toxins that perhaps keep things from eating them. But these natural products are are special metabolites. And so here I just sort of try to define for you what natural products are. Most of us understand biochemistry and the role of amino acids and sugars and fatty acids and the like. And so those are all metabolites that are involved in primary metabolism. So natural products is this group of chemicals that are made by living organisms, just like amino acids and fatty acids and the like, the primary metabolites, but they're what we refer to as secondary metabolites. And they're secondary primarily because they're species specific. So you don't find the same secondary metabolites generally in two different species. They're highly diverse biomolecules. They're not really related in a primary fashion to one another. They are related by their biosynthetic pathways. And so here I've defined natural products as these post-translationally modified oligomers of isoprene and acetate and amino acids. So they have the same building blocks, but it's this biosynthetic process, in fact, that Nicole is going to talk about in her presentation, that defines them as secondary metabolites or natural products. These natural products, as I alluded to earlier, are produced by these organisms for ecological success, as I mentioned, as antibiotics, as toxins, sometimes as pheromones. We chemists are often interested in these things for their applications. So we find natural products a lot in pharmaceuticals. I'll talk a little bit more about that in a minute. We see them in consumer products such as food, cosmetics, fragrances. And this is probably where most people interact with natural products. And I sort of highlight that in this slide as showing you some natural products that perhaps you interact with almost on a daily basis. But again, to emphasize, you know, the vanilla bean doesn't make vanilla to flavor our ice cream. It makes vanilla to attract pollinators and the like, right? So these are natural products that have ecological roles for the producer that we have then found applications for, often in daily life. Some of the things that you will also be familiar with natural products include the likes of pharmaceuticals, and I'm showing here just several that you're perhaps familiar with, certainly penicillin and aspirin. These things come from living organisms. Aspirin, the willow tree, penicillium mold makes penicillin. And I'm going to call out Taxol right now. So this is an anti-cancer agent that's only approved for clinical use within the last couple of decades. It's a fairly new anti-cancer drug. But this is one of the things that I think really highlights why chemists are interested in the microbiome. So it was almost 20 years ago that Gary Strobel and the Steriles found that Taxol, which had originally been isolated and characterized as a metabolite of this yew tree, they found that the yew tree produced fungi. So you see an endophytic fungus growing out of the tissue of the tree here that's placed on a petri dish. That fungus actually produces the Taxol. So the anti-cancer drug that we had thought was produced by a tree, a eukaryote, well, fungi are eukaryotes too, but right, the microbiome includes the likes of prokaryotes, and we find that in fact so since 1990, since really the invigoration of this field from this discovery of Taxol in Texas in the yew tree, there's been a number of microbiome studies. Actually, I'll say there's been a lot of microbiome studies by now. We've found, in fact, that a lot of natural products that we thought were made by trees or plants or sponges or corals are actually made by microorganisms living within those tissues. And so the story that we're going to tell today, that Nicole primarily is going to tell, is how we've advanced this project with Palmerolide, with the idea that perhaps it was microbially produced. This is where Alison Murray came into the project, and ultimately Patrick Chain. And I will let Nicole take over from there and tell you what we now know about Palmerolide and whether it's a microbial natural product or a tunic, Ascidian natural product. Nicole?
Can you see my screen okay, or is it having the same issue with the green bar?
I see it.
Okay, great. So yes, this has been a wonderful project to be a part of, and I'm very excited to share with you guys what our workflow has been and what we have found so far, and the process of how we've gotten there. So like Dr. Baker said, this is a multi-decade project that began with the collection of Synoicum adoranum from Antarctic waters in the early 2000s. And it brings us to the present day. So the overview of our workflows goes from the sample collection to isolation of a bioactive molecule with a goal of establishing a pathway to drug development. So this branches out to larger scale extraction, to synthetic routes, and then to defining the biosynthesis and microbiome genomics. So nature is a rich source of biologically active molecules. So over 60% of our FDA approved medications are directly from nature or inspired by nature. These molecules are often secondary metabolites, like Dr. Baker mentioned, that are produced to enhance the survival of the slow or sessile organisms. One class of compounds that demonstrates immense pharmaceutical potential are polyketides, which consist of hydrocarbon chains with varying degrees of oxidation. These are biosynthesized by large mega enzymes that are similar to, but evolutionarily divergent from, fatty acid synthases. So the molecular machinery has been hijacked and enhanced to create beautiful and architecturally diverse molecules with strong affinities for biological targets. So polyketides show bioactivity as cholesterol-lowering agents and as antimicrobial agents by disrupting ribosomal function. They also have effects as anti-tumor agents, inhibiting angiogenesis and cell proliferation, inducing apoptosis, and altering the eukaryotic cytoskeleton. And you heard that we work with Antarctic organisms. The Southern Ocean is a unique environment. It has seasonal extremes in the marine ecosystem in regards to ice cover, light availability, and food resources. The effects of glaciation and continental drift also contribute to speciation. So in addition, the Antarctic circumpolar current isolates the waters of Antarctica from the remaining oceans. So these factors result in rich biodiversity, and the competition that results within this biodiverse ecosystem leads to great chemodiversity. So in 2006, the Baker Lab Group published Palmerolide A, which is a marine macrolide polyketide isolated from Synoicum adoranum, as Dr. Baker mentioned. So it showed efficacy against melanoma skin cancer cells with a high degree of selectivity. And it's known to act as a VATPase inhibitor and to induce autophagy. So Synoicum adoranum is georeferenced to the circumpolar waters of the Southern Ocean and to the Alaskan coastline. So each colonial sea squirt is comprised of hundreds of zooids. And these ascidians are known to be rich in secondary metabolites, the palmerolides, the ectosteroids, the ceramides. They are also known to possess a rich and diverse microbiome. So Palmerolide A has strong pharmaceutical promise, but it's not ecologically or logistically feasible to mass harvest the organism. And multiple synthetic routes have been outlined in the literature, but have not established an efficient route for drug development. So our goal through this project, and part of my dissertation work, is to identify the biosynthetic machinery to then allow us to move forward with the next goal of heterologous expression. So biosynthetic gene clusters are discrete genomic packages that encode for mega enzymes that serve as molecular assembly lines for natural products. The discrete genomic packages can be horizontally transferred from one organism to another. In addition, based on evolutionary pressures or streamlining, modules can be lost, gained, or their functions can be altered to form new secondary metabolites that will aid in survival. So these can not only be transferred from bacteria to bacteria, but there's evidence of horizontal transfer between prokaryotes and eukaryotes even. And they can be found as plasmids, but are often integrated into the genome of the organism. So with mass extraction and synthetic routes out, the next step was to determine how to find the genomic information for this biosynthetic machinery. Could we find a microbial producer and then sequence the genome? So it's known that many secondary metabolites isolated from marine invertebrates are actually the product of a complex symbiosis between the microbes and their eukaryotic host. And in the genomic age, this is becoming even more clear. Microbes from the marine environment are very difficult to culture, with over 99.9% of them being deemed unculturable in standard laboratory conditions. Early attempts to find the true producer of the Palmerolides, and therefore the biosynthetic gene cluster, were culture-based. 16 bacterial isolates were successfully characterized from Synoicum adoranum in the hunt to find the Palmerolide producer. So this would be the gateway for an ongoing supply of the compound. Most of them were from 5 genera of gamma proteobacteria, and one was from an alpha proteobacteria. The liquid cultures from all 16 isolates were used to screen for the presence of Palmerolide A based on liquid chromatography techniques, but none revealed the presence of Palmerolide A. Sometimes biosynthetic gene clusters can be silent, and one genome from the cultivar library was sequenced, this pseudovibrio species seen here. So the genus is known to be very rich in biosynthetic gene clusters, but this was the first one isolated from Antarctica, and it was quite exciting for the team. It has an interesting phenotype and an interesting branching division pattern. We were able to obtain the full genome sequence and can see that it's also very biosynthetically talented with a slew of biosynthetic gene clusters, but it is not the producer of the Palmerolides. The next phase of the project delved into the microbiome genomics first for the answers. Our goals continue to be, number 1, identify the Palais biosynthetic gene cluster, and number 2, identify the microbial producer, but this time with a fresh strategy. So we used a metagenome sequencing strategy with 454 and ion torrent proton sequencing for the first goal. And for the second, we used ITAG sequencing of the variable 3,4 regions of the bacterial 16S ribosomal RNA, followed by Illumina amplicon sequencing. The first step to identifying the biosynthetic gene cluster was to develop a retrobiosynthetic scheme. We take a look at the molecule, decide what class of enzymes would be responsible for its biosynthesis, and then dissect each part to decide which specific enzymes would contribute to the architecture. Here we have a compound that is classified as a polyketide with a peptide component to incorporate the nitrogen. If we look at the subtypes of polyketides, there are 3 subtypes. Type 2 is an iteratively or repeatedly acting enzyme machinery that has few enzymes that act in a repeated manner to elongate that polyketide chain. Type 3 is also iterative or repeatedly acting based on homodimeric units with sequential condensation and elongation of the polyketide chain. Those often result in aromatic products. Type 1, however, is non-iteratively acting, with each module creating a specific step, and that leading to elongation of the polyketide chain. So we chemists like to put these enzymatic domains into little balls. So that's this representation. You'll see an image of what it looks like in the crystal structure in a bit. But this is an overview of the type 1 Pks systems. There are 2 subgroups, those that house their enzymes for subunit selection within the megaenzyme, and those are called cis-AT, or acyltransferase, and those that have selector enzymes that are encoded outside of the core biosynthetic genes called trans-AT Pks systems. And these systems, collinearity is not the rule, meaning that there can be parts of it that are encoded in other places that have actions that either add to or augment that architecture at that point. And this allows for great architectural diversity. So here, Ks is a ketosynthase, DH is a dehydratase, ER is an enoyl reductase, and TE is a thioesterase. So we had suspicions that a trans-AT type 1 Pks system was responsible for the biosynthesis of our compound of interest. So this is a video from the University of Michigan that shows the circular or modular representation of a biosynthetic gene cluster, as well as crystal structures of a portion of a polyketide synthase. So since the video audio is difficult to stream over Zoom, I'll give you the rundown of the scientist, of what the scientist is saying. So what we see is the biosynthetic pathway of the polyketide. It's a multi-enzyme factory that sequentially extends the carbon chain. The factory is divided into modules which contain multiple domains. The orange circle is an acyl carrier protein, and it works to transfer intermediates from one module to the next. The fifth module is shown here with the various portions via cryo-EM. In blue, we have the ketosynthase. In green is the acyl transferase. In purple is the keto-reductase. And the acyl carrier protein is in orange. So this transfers the building blocks from the acyl carrier, the acyl transferase to the ketosynthase. And then the intermediate to the keto-reductase. And finally to the next module. The upstream ACP transfers the intermediate to the ketosynthase, here seen in red. The ACP of this module gets a building block from the acyl transferase here in a cis-AT system. You can see that it's integrated with it rather than free floating. And then the ACP carries the building block to the ketosynthase for the elongation reaction. It then transfers it to the keto-reductase for reduction of the beta-carbonyl, and then onto the next module. So this is just one portion of the mega enzyme that's used for Pks synthesis. So it's quite an exquisite system. So polyketide biosynthesis typically occurs in this assembly line fashion through mega enzymes with many catalytic domains. So to summarize the biosynthesis for trans-AT type 1 Pks systems, there are 4 basic steps: initiation, elongation, alteration, and termination. So initiation occurs when an acyl transferase selects a starter unit, and this starter unit is tethered to an acyl carrier protein via a thioester bond. The next subunit is then selected and transferred to another acyl carrier protein. The ketosynthase catalyzes the Claisen-like condensation to create an elongation of the growing chain by 2 carbons. The beta-carbonyl may then be modified through tailoring enzymes to alter the oxidation state. In addition, a methyltransferase can add a methyl group in the alpha carbon using acetylmethionine methionine as a substrate. Progressive elongation and modifications occur until the termination step, where a thioesterase works to release the molecule in linear form or to cyclize and then release the molecule. Additional enzymes that work in trans to the mega enzyme can further modify the compound. So pencil on paper, Dr. Baker drew out some of the predicted components several years ago. I just love it. With all his brilliance, Dr. Baker was able to scribble down a synthetic plan at the enzymatic level in the early stages of polyketide synthase mega enzyme knowledge. So look at this masterpiece. And one of the first components of my role in the project was to expound on the initial retrobiosynthetic prediction. So there are several key biosynthetic features, including an NRPS-PKS hybrid system with a PKS starter unit, followed by incorporation of a glycine subunit, likely through a non-ribosomal peptide synthase pathway, followed by 11 rounds of polyketide elongation with various modifications. There is an alpha methylation through methyl transfer from acetylmethionine methionine to create the C26 and C27 methyl groups. In addition, there's a pendant carbon installed in the beta position, likely arising from HMG-CoA synthetase. HMG-CoA synthetase has been seen in multiple marine macrolides of microbial origin, such as Jamaicamides, bryostatin, curacin, and oocytin. So here's a further indication that this polyketide is produced by a bacterium rather than its eukaryotic host. The carbamate installation was expected to be a result of a carbamoyl transferase, and the C10 hydroxyl group was hypothesized to form a hydroxylase or from incorporation of a hydroxylated subunit. So with the detailed retrobiosynthetic prediction in hand, we proceeded forward. The Ascidian tissues were homogenized and the eukaryotic and prokaryotic cells were separated from one another. The bacterial pellet was used for analysis. The metagenome was then sequenced by Dr. Alison Murray using 454 and ion torrent proton techniques. After assembly of the 17 gigabases of DNA by our collaborators at Los Alamos National Labs, the data was run through anti-SMASH for annotative screening of biosynthetic gene clusters. Many biosynthetic gene clusters were identified, but one was of particular interest because it had great congruence with our retrobiosynthetic strategy and was contained on a single assembled contig. The annotation reveals a hybrid Pks NRPS system, trans-AT in nature, an HMG-CoA synthetase, and a carbamoyl transferase. Using a combination of the annotations from anti-SMASH and manual alignments, I've worked on delineating each step of the mega enzyme assembly line process for Palmerolide A. So anti-SMASH is a wonderful tool to get us started on the journey of specifically annotating biosynthetic gene clusters, but mysteries arise, especially when trans-acting polyketide synthases are at play. So we had several of these wonderful surprises pop up during the process of elucidating the gene cluster. And one such was the reduction of a couple of bonds to fully saturated hydrocarbon segments. So typically there's an enzyme called an enoyl reductase present in line to perform this action. However, in ours, it wasn't. We discovered that there was a precedent for these being encoded as transacting enzymes, even outside of the biosynthetic gene cluster. And this is what we proposed for our cluster. In addition, if you remember, we talked about a thioesterase being involved in the release of the molecule from the mega enzyme. But there was no thioesterase at the end of our core biosynthetic genes. But there was a truncated condensation domain. Two other Pks systems cite a termination condensation domain. Our condensation domain, though truncated, maintains much of the HHXXXDG motif, most importantly the second histidine, which serves as the catalytic histidine in the condensation reaction. So this shows conservation of the motif when compared to other termination condensation domains and with the protein family hidden Markov model seed that is used for training the PFAM algorithms. Once the thioester bond is cleaved through cyclization, transacting enzymes are at play. So for example, a transacting hydroxylase installs an alpha hydroxy group that's key in the bioactivity of Palmerolide A. The carbamate group is then transferred to form the final product. So now we have identified the biosynthetic gene cluster, which was an exciting accomplishment, but what about the producer? The biosynthetic gene cluster was hanging in genomic space with no phylogenetically conserved genes to inform us or point us to a producer. We know that biosynthetic gene clusters are often horizontally transferred, so it's not necessarily a surprise, but we had to use a strategy to address this. So we decided to delve into the microbiome. Our team used a nested sampling approach. 7 sites from the boating area around Palmer Station, Antarctica, were chosen. During the dives, 3 colonies of the Ascidian were selected from each site. A colony looks like this. There are multiple lobes comprised of a stalk and a head that form a colony. Hundreds of zooids make up each lobe, so a single lobe represents hundreds of animals. 3 lobes from each colony were then collected. This degree of biological replication is unprecedented in Antarctica. It also is quite different from the typical sample collections for natural product discovery. Typically, multiple organisms are batched together for compound extraction and analysis. So here's an example of how the Ascidian samples were collected. So first, the metadata. Note that temperature, 1 degree Celsius. Dr. Baker and his dive buddy are outfitted in dry suits to keep them adequately warm. But dexterity with thick gloves is pretty difficult. So this is really incredible. And note the high degree of biodiversity seen in these Antarctic waters. There's bryozoa, soft coral, encrusting sponges, red algae, green algae. They're all in the vicinity. So Dr. Baker collected water samples near the organism of interest before collecting the lobes of the Ascidian. He gently pulled at the invertebrate, obtaining the holdfast, stalk, and the top of the lobes. This procedure was followed for all 21 collections, 3 at each of the 7 locations. So this careful sampling was designed to look for the pattern of chemistry across 63 samples, with the ability to analyze lobe-to-lobe differences and colony-to-colony differences in the chemistry and in the microbial communities. The lobes were frozen and then divided in half. The first half of the lobe was used by Sophia in LCMS analysis, specifically looking for Palmerolide A. The other half was used for the microbiome work. So all samples had significant levels of Palmerolide A. It's the main secondary metabolite found in the organism. So liquid chromatography, mass spectrometry was used to confirm the presence of Palmerolide A in all samples. In the top left panel, you can see the chromatogram with a large peak just past 600 scans. The mass spectrum obtained of the compound that eluted at that peak shows an M/Z of 585 correlating with the M+H, 607 correlating with M plus sodium, and 567 correlating with M minus water plus H adducts. The graph on the upper right shows lobe-to-lobe differences in Palmerolide A levels. Triangles here indicate significant colony-to-colony differences within that site, filled triangles with significant differences from other two colonies from that site, and open triangles with significant differences from one of the other colonies at that site. The second half of the lobe was used for the microbiome work. ITAG sequencing was performed based on 16S data to determine the amplicon sequence variants. Since ASVs roughly represent different species or strains, you may hear me refer to them as species moving forward for simplicity's sake. So when we looked at the structure of the microbiome for Synoica we decided to follow a precedent that was set in a paper published by Hernandez Agreda and her colleagues who study the microbiomes of corals in Australia. The core is defined as those ASVs with persistence across at least 80% of samples. Since the depth of ASV sequencing was subsampled to only 10,000 for each of our samples, a cutoff of 80% seemed to be very reasonable. In fact, the members of our core all had persistence across 90% or greater. The dynamic represents those species with presence between 50 and 80% of samples, and these may serve key metabolic roles, but may be ecologically interchangeable with another species. The variable species show presence in less than 50% of samples. The variable members represent those ephemeral species that are environmentally stochastic and may represent food source for the Ascidian or a passerby in the water column. Interestingly, the core members only comprise 21 of the 462 species identified, representing only 4.5% of distinct ASVs. However, when we look at the total number of occurrences, we see that the core members represent nearly 95% of all occurrences. On the flip side of that, the variable members are over 92% of the distinct ASVs, but only make up 3.3 of the total occurrences. All but one member of the core were distinct from bacterial plankton datasets from multiple field seasons. So we wanted to look further at the structure of the microbiome and we decided to use the Primer-E software package, which is a relatively user-friendly statistical tool for ecology. First, we wanted to create MDS plots to visualize our data. So here we see the metric multidimensional scaling plot of the core microbiome based on site. Each site is notated by a different color and symbol. So based on the composition of our data, we decided to use a Bray-Curtis similarity matrix of square root transformed ASV occurrence data to create the TM-MDS plots, which are metric multidimensional scaling with no constraint to the y-intercept. In addition, we bootstrap the data to improve visualization of differences. Each site-based data cloud is shaded and the centroid is demarcated by its respective black symbol. The same analysis was run on the dynamic subgroup and on the variable subgroup. Here we see the TM-MDS plot of the complete microbiome of all 63 samples. Each site is notated by the different color and symbol, and visually we can see some separation of the microbial communities based on site. So for example, the samples from Janus Island, which are the turquoise squares, seem quite distinct from Dalaka Island, which are the blue triangles. We also see that the data cloud for killer whale rocks, which are the seafoam diamonds, are tightly clustered, while others are more dispersed. The pairwise comparison between colonies at each site shows coherence and a high degree of similarity for this microbial community. So the pairwise similarity between colonies is between 54 and 88%. And what explains this? statistical analysis using Permanova, a permutationally derived multivariate analysis, was performed. The components of this nested experimental design, site and colony, can be evaluated as drivers of variability. The component of site-to-site variability explains roughly 25% of the total variability in the microbiome. Colony-to-colony variability explains 28%, while the remaining 47% of variability is unexplained or stochastic. We also looked at the dispersion of data clouds around the site-based centroids and found a trend that pointed to further coherence in the core. As expected, the coherence in the core was much more tightly centered around the centroid when compared with the dynamic group, which was less dispersed than the variable group. So here you can see the average for the core ASVs was around 9 across all sites, increasing to 28.5 for the dynamic and 54.7 for the variable. So remember that our second goal in the project was to identify a potential producer. So let's take a different look at the members of the core. So this is the heat map of square root transformed amplicon sequence variant occurrence data of the core microbiome with hierarchical clustering based on the sample composition seen in the x-axis. The ASVs are ranked and numbered and shown here on the y-axis. Nodes with significant clusters are indicated from left to right, and the ordering of clustering inside the node was not significant. So the horizontal line drawn below SAM ASV 16 here, which is Synoicum adurinum microbiome, ASV 6, demarcates the cutoff of ASVs that were present in all 63 samples. This was using the DADA2 data. The pattern seen in the heat map is very interesting in regards to ASV 15. So as a member of the core, we know that it has ASV representation in at least 80% of samples, but there's a notably high amount of this species in some samples, but not in others. So Dr. Allison Murray did some beautiful genomic correlation calculations and was able to determine a putative producer, which led us down a road of confirming this finding. So this will serve as a bit of a teaser here because we're working to publish these findings and the manuscript is in prep. So binning of the 16S rRNA gene data that we've acquired from the metagenome echoes the findings of our 16S rRNA gene I-tag sequencing. So here's a sunburst showing that composition. And if we overlay the genus and species information from the ASVs, we'll see a similar breakdown as shown here. So this further confirms our microbiome composition findings. And back to that promising lead on the Palmerolide producer. So additional iterations of sequencing were performed and the genome was polished by our collaborators at Los Alamos National Laboratory, a team known as the Genome Finishers. And so we were able to achieve the assembly of a MAG or metagenome assembled genome. So this genome represents a new genus and species and we're excited to share this data in an upcoming publication. So the next major goal is heterologous expression of the biosynthetic gene cluster, which would pave a path for drug development of this pharmaceutically promising macrolide. So our workflow from collection of invertebrate to isolation of bioactive molecule to paving a path of drug development using microbiome genomics has brought us to the biosynthetic gene cluster and a putative producer that we are sure actually houses the biosynthetic gene clusters now. So in conclusion, a genomic approach can inform the chemistry and pave the way for drug development for lead compounds from marine invertebrates and of other natural products with pharmaceutical promise. So with that, I'd like to thank Dr. Baker for allowing me to work with him on this exciting project and our entire lab group, including my wonderful undergraduate researchers. I'd also like to thank our collaborator, Allison Murray at DRI, who's been the microbial ecologist bridge between the realms of genomics and chemistry. She's also been a great personal mentor to me. Patrick Chain's team at Los Alamos National Labs has solved so many great genomic challenges. I'd like to thank the funding agencies that have made this work possible, the NIH, NSF, DRI's IPA program, JGI, and the Office of Graduate Studies here at USF. And I'd also like to thank you for your kind attention and would welcome any questions that you may have.
Thank you so much, Bill and Nicole. And it's always nice to hear research that has a very happy ending. And we'll look forward to the publication. I sort of know the answer already, but I won't reveal, I promise. And so you have a choice. You can either jump right in if you have a question or post something to the chat. So There is one from Zilin. How did you separate the bacterial cells from the homogenized tissue?
So this was through differential centrifugation, but if you want more information, we can get Allison, if she's here, I see her, to give a little more information.
Oh, thanks for unmuting me.
You're welcome.
Yeah, so we've used a couple of different types of homogenization, but the bacterial cells and the host cells are so different in size that once the homogenate is prepared, we can go through rounds of centrifugation to pellet the host debris and keeping the bacterial cells in suspension, and then we pellet the bacterial cells. We also, the details of that are also in the publication that Nicole was referring to that just came out in Marine Drugs last week. So that's described there in a little further detail.
Okay, other questions? Somebody's typing a message, I think, maybe not. Other questions? Going once, going twice. Everything was clear, Nicole, and it's great work. And you have in the chat several people saying great presentation. So thanks to both of you. I think we will just sign off, and all the best to everybody. Enjoy the rest of your day.
Thanks, Wayne. Thanks everybody for chiming in.
Thank you.
Bye-bye.
Bye, everybody. Take care.
Great.